Hi, everybody. We know that the passage of time is a constant, but the actual process of biological aging can look very different from one person to the next. In this explainer, we're going to take a look at a 2025 study by Wang and colleagues. They investigated the relationship between two fundamental drivers of aging, the decline of our mitochondria and the subtle, age-related changes to our genome's expression, what we call epigenetic alterations. So we'll be exploring how these two distinct biological clocks might actually be connected.
Here's how we'll break it down. First, we'll define the core concepts of chronological and biological age. Then, we'll get into the study's design. After that, we'll examine the two main findings, one on rare mitochondrial mutations and the other on mitochondrial abundance. From there, we'll explore the link to metabolic disease and finally wrap up with the broader implications of this research.
Okay, first things first, we need to establish a foundational concept, which is the distinction between our age in years and the much more dynamic state of our physiological age.
So chronological age is simple. It's just the number of years you've been alive. Biological age, on the other hand, is a measure of your body's functional state. It reflects the cumulative impact of molecular and cellular damage, and it can vary quite a bit between people of the same chronological age. It really is a more accurate indicator of overall health and mortality risk.
So how do we measure biological age? Well, one of the most robust ways is through epigenetics, specifically by analyzing DNA methylation patterns. These are the chemical tags that regulate gene activity. Using these patterns, scientists have developed what we call epigenetic clocks. When the age predicted by one of these clocks is higher than a person's actual chronological age, we call that epigenetic age acceleration, or EAA. A positive EAA means you're aging biologically faster than expected.
The second key hallmark of aging we're concerned with here is mitochondrial dysfunction. We can assess mitochondrial health using a couple of key metrics. The first is mitochondrial DNA copy number, or mtDNA-CN, which is a proxy for how many mitochondria are in your cells. The second is heteroplasmy, which describes the presence of different mtDNA variants within a person. This can include both inherited variants and new somatic mutations that build up over a lifetime.
And this leads us to the study's central question. Do all these different aspects of mitochondrial health relate to epigenetic aging in the same way? The investigators hypothesized that they don't. Their thinking was that the burden of rare somatic mutations in the mitochondria would have a very different relationship with epigenetic age compared to, say, common inherited variants or just the total number of mitochondria.
So, to test this hypothesis, the researchers put together a comprehensive study using multiple cohorts. Let's dig into their methodology. They leveraged data from over 6,300 participants across 4 large long-term studies. This gave them a statistically powerful and diverse sample, with a wide age range and representation from both white and Black American participants. That diversity is important for the generalizability of the findings. Their analytical framework was broken down into 4 main steps. First, they used whole genome sequencing data to quantify those mitochondrial metrics we talked about, the copy number and the burden of rare and common variants. Second, they calculated epigenetic age using a whole suite of clocks, from the early ones to the latest generation. Third, they ran a meta-analysis to look for statistical associations between the mitochondrial and epigenetic data. And finally, they used mediation analysis to probe potential causal pathways, linking all these factors to disease. Okay, so that's the setup. Let's turn to the results. The study's first major finding has to do with the impact of rare mutations in mitochondrial DNA. What the analysis revealed was that a greater accumulation of these rare, potentially harmful mitochondrial DNA variants was positively correlated with accelerated epigenetic aging. Interestingly, this association was strongest with the first-generation clocks, Horvath and Hannum. Let's put some numbers on that. The meta-analysis showed that for each 1 standard deviation increase in this rare variant burden score, biological age went up by 0.20 years on the Horvath clock and 0.22 years on the Hannum clock. It's a consistent, measurable link between accumulating these mutations and having an older epigenetic profile. From a statistical standpoint, this finding was quite robust. The p-value for the association with the Hannum clock was 1.3 times 10 to the negative 6. That indicates a very low probability that this result is due to random chance, supporting a solid relationship between these rare somatic variants and this measure of biological age.
Now, here's a crucial distinction. This association was specific to the rare variants. When they looked at the burden of common, largely inherited variants, there was no significant association with any of the epigenetic aging metrics. This is a key insight. It strongly suggests that it's these somatic mutations that accumulate over a lifetime, not necessarily what you inherit, that are linked to the acceleration of this epigenetic clock. They also noted this effect was more pronounced in people over 60.
Okay, that was the first finding. The second major finding shifts focus a bit, moving away from the quality of mitochondrial DNA and towards its quantity.
In this case, the study found an inverse relationship. Individuals with a lower mitochondrial DNA copy number, meaning fewer mitochondria per cell, actually showed faster biological aging. This association was specifically picked up by the more advanced later generation clocks like Grimmage and Dunedin-Tace, the ones that tend to be more predictive of health outcomes. The meta-analysis quantified this. A 1 standard deviation increase in MTND copy number was associated with a 0.11-year deceleration of Grimmage. It was also linked to a decrease in the Dunedin-Pace score, which is a metric for the current pace of aging. So the negative coefficients here just mean that a greater abundance of mitochondria is associated with slower epigenetic aging, at least according to these particular biomarkers. So, building on these two findings, the next logical step for the researchers was to ask if this interplay has any clinical relevance. They decided to look at it in the context of metabolic disease. It's fairly well established that lower mitochondrial abundance is a risk factor for things like obesity and type 2 diabetes. The researchers wanted to test a specific hypothesis for why that is. They proposed that epigenetic age acceleration might be a mediator, a sort of mechanistic go-between that connects the dots from reduced mitochondrial numbers to these adverse metabolic outcomes. The model they tested had two possible pathways. There's a direct path, where lower mtDNA copy number just independently increases disease risk. But there's also an indirect, mediated path, where lower mtDNA copy number first accelerates the pace of aging, measured by Dunedin-Pace, and that accelerated aging is what contributes to the higher risk of disease. And the mediation analysis provided some evidence for this hypothesis. In the Framingham Heart Study participants, the pace of aging, measured by Dunedin-Pace, was found to mediate about 11% of the total effect of mtDNA copy number on obesity risk, and just under 11% of its effect on type 2 diabetes risk. This suggests that roughly a tenth of the mechanism linking mitochondrial abundance to metabolic health seems to operate through this acceleration of the biological aging process. All right, let's synthesize these results and consider what they mean for the broader field of aging research. So there are a few important conclusions here. First, the buildup of rare somatic mitochondrial mutations appears to be a distinct biomarker of biological age, separate from inherited variants. Second, the authors suggest that mtDNA copy number might not be a direct driver of aging, but more of an indicator of systemic inflammation, because its association with epigenetic aging was reduced after they adjusted for cardiovascular risk factors. And third, the study provides evidence for a specific mechanism, that accelerated epigenetic aging is a partial mediator connecting lower mitochondrial abundance to metabolic disease. Now, when interpreting these findings, it's also important to consider the study's limitations. They had to exclude variants with an allele fraction below 5%, which means they might have missed some true low-level mutations. The design was also cross-sectional, which is great for finding associations, but it can't definitively prove causality. There were also some technical differences across the cohorts and some missing genetic data that constrained certain analyses. What this research really does is illuminate the complex communication happening between the mitochondrial and nuclear genomes as we age. And it leaves us with a critical question for future research, a sort of chicken or the egg problem. Do these rare mitochondrial mutations directly cause epigenetic dysregulation? Or are both of these things just parallel consequences of a more fundamental upstream decline in our cells' quality control and maintenance systems? Answering that question will be fundamental for developing interventions that can truly target the core mechanisms of aging.